So first, I want to bring you into the world of Nomo, which is where I have my experience, where we are looking at the genotyping and the phenotyping a lot. So when you look at genotyping, the genotyping is only detecting the cell wall polysaccharide genes, which is the DNA sequences and the potential functions of the bacteria. Within the vaccine relevance, it is showing the predicted serotypes, which is potential, where if you go to the phenotyping method, this is actually detecting the expressed capsular polysaccharides, and it's detecting the active expressed genes and also the observed characteristics, behavior of the bacteria. When it goes that, if we then look at the vaccine relevance of phenotypic method, this is directly affected the vaccines. It is antibody binding, and it's also what you would show when you're seeing in the OPA, the killing assay. So this is why phenotypic method is important. If we then look at the strengths of the two methods, so genotyping, I guess, again, is really high sensitivity and specificity. You don't depend on the growth condition of the bacteria. And it's also a scalable and high push-through method. It's also the only method which can actually guide you for potential new phenotypes. On the other side, phenotyping method is directly affecting the immune recognition of the bacteria. It's also the golden standard within serotyping, which is the Quilling reaction. If we look at the weaknesses of the two methods, when you look at genotyping, it doesn't guarantee expression of the genes. It can only be used in the vaccine as a inferior part for relevance. It's you need skilled techniques for doing it, tools and equipment. Within the phenotyping method, this is really relying on sensitivity of the growth patterns and the operators during the method. It has a low push-through, and it's also subjected for interpretation. You need skilled people to do the Quilling reaction. So this also brings me to why is this for the phenotype critical during the vaccine development? So this is lessons learned from the pneumococcal world, where vaccines are targeted against the expressed antigens to capture the polysaccharides and not the potential genes. The risk when you rely on only the genotyping, it is that you will not see what's actually expressed by the bacteria, but only what could be expressed. So you wouldn't have the recognition and the killing effect of the bacteria. And when you look at phenotypic expression, this is actually not just showing what the genes are present, it is also showing the actual protection. But one thing is very important is that you need both methods. It's very important to do genotyping and phenotyping because you need to have the accuracy of your data. You need to have the best surveillance data, the best research within pneumococcal bacteria. And by doing both methods, you see the already recognized serotypes, but you also get to see potential new serotypes, which is popping up because of the vaccines. And this has been the approach within the pneumococcal world for a lot of years. Within pneumococcal, you are doing both the genotyping method, and then you will screen with the phenotypic method afterwards to ensure that it's actually expressed in the capsule. But what would then happen if we look at new and emerging areas? Klebsiella pneumonia is been recognized by WHO as a new potential vaccine candidate because of the high antibiotic resistance within the bacteria. And then we look into the surveillance data, which is really important when you need to develop a vaccine. You need to be able to see what is actually shown in the surveillance data so you can develop your vaccines against the right serotypes. And how we decide on this? For the genotypic data, it should be shown as a K-type with an L after for locus. And for the phenotypic data, it should be defined as a K serotype. But what is actually happening in the data which is being presented for the world is that the genotypic data is a mixture of the two nomenclatures and are including both the K locus type and the K types. This was a big study which was presented at the first Klebsiella meeting in Paris in 2024. It was presented by Statens Serum Institute and was actually funded by Bill Gates. And in this study, they were looking at 299 clinical samples. They used two methods, which were genotyping using whole genome sequencing method, and then they were using the golden standard phenotyping method, which is counter current electrophoresis method, where they use polyclonal antisera. And this is the data. And it actually showed that when you looked at the genotyping and the phenotyping, there was a mismatch of 41% for Klebsiella pneumonia. And if you looked even further, you could see that 80 samples were mistyped to be new serotypes, but it was actually the current serotypes, which was already defined by the phenotypic method. Furthermore, you could see on this picture that we had 4 new serotypes coming up with high virulence. There was 94 samples, and out of those, only 50 of them were actually known serotypes. The poster result was showing that half of the serotypes was characterized as new genotypes, but it was actually the current phenotype. And this makes it really important that you always check your genomic data with phenotypic methods. It also showed, like I said, the 4 most prevalent new genotype showed identical capsular types. So KL102 was actually K18 if you looked at it in the phenotypic method. And for KL112, it was actually a K24 type. And KL122 was K35. And the last one was KL136, which was a K61. So during this symposium, there was also vaccine candidates being presented during the meeting, which actually included the KL102. So if we look at that, they were saying that it was a KL102 genotypic type, but they were also thinking it was a new phenotype. But these data actually showed there was a K18 instead. So by not doing your phenotypic method, we are presenting the vaccine world for these wrong data, and there's a risk of reducing your specificity when the vaccines are designed by covering multiple overlapping K-types. So in conclusion, in the poster, it is that you need to combine the two data sets. You need to do your genotypic data, and you need to do your phenotypic test as well to ensure that it is actually novel capsule types and not current capsule types. So when going back and looking at this as a total, you can say that what is critical for a vaccine development is that you have your infection, you're doing your surveillance data. So this is an example for Klebsiella and pneumococcal. You do your surveillance, so after you have the infection, you send in your sample to the surveillance lab. They will do either screening on whole genome sequencing, and then afterwards they need to do a control of the genotype to see if it's actually a new phenotype or if it's a current phenotype. If, in case the genotype is not matching the phenotype, it is a potential new serotype. It's important you don't do monoclonal for this because you want to see an actual immune response. So you have to actually do it in rabbits and produce monoclonal antisera to see that it's actually inducing a true immune response. When that is done, then you're ready to share your data with the vaccine industry. So you ensure that all your data is actually true phenotypic types and are producing an immune response, which is relevant when you're developing your vaccines. So in conclusion, this goes for all bacteria. You need to combine your genotypic data with your phenotypic data if you want to rely on vaccines which is actually producing an immune response in patients. So that was the conclusion. Thank you so much.